The process of photosynthesis requires few raw materials, and in order to judge that is it necessary that the photosynthesis actually needs those materials, we are performing few experiments. So the next experiment which we will be performing will make us understand the importance of light and how light is important for the process of photosynthesis. So the aim of the experiment is to show that light is necessary for photosynthesis. The materials required remains the same, just an addition is the Ganong's light screen or the black paper strip. So anything which is black can be used, even a black cloth can be used in order to do this experiment. We just need to cover the leaf with the help of that black cloth. So there is one idea in mind with the help of which we perform this experiment. First thing is that light is required for the process of photosynthesis. Why is it required? Because it activates the chlorophyll molecules, and after the activation, the electrons are released. Those electrons play a very vital role in the entire process of photosynthesis. Basically, the initiation step of photosynthesis is because of the presence of light. So, the idea is when a part of a leaf is covered from both upper and lower side with a black paper strip, at that time light will not reach that covered area, and hence there will be no photosynthesis in the covered part of the leaf. Whereas the other sides of the leaf, the area which is uncovered, will do normal photosynthesis. So when we do the iodine test, or we want to check if glucose is present or not, then we will be able to find out that glucose is only present in the area which is uncovered, whereas the covered area will not show the presence of glucose. Now, the glucose which is formed will carry out most of the metabolic activities, and the rest of the glucose which is formed will be stored in the area itself. And that's why the iodine test will be positive for the uncovered area of leaf, whereas the covered area will be showing no iodine test or negative iodine test. You can see the plant. We are not going to uproot or take out this small leaf from the plant. We will just let the leaf be covered with the help of this black paper strip once the destarched plant is brought out of the dark place. So as soon as the dark plant or destarched plant is out, how do we destarch a plant? We destarch a plant by keeping it in dark for 48 to 72 hours, and then after, we take the potted plant, cover one of the leaf with a black paper strip, and then place it in the sunshine for 48 hours. Sorry, it's not 48, it's 2 to 3 hours. So after putting the plant for 2 to 3 hours in the sun, what we see is the plant has started doing photosynthesis. And then after 2 hours we will take out the, or we will pluck that leaf which is having that paper strip covered on the upper as well as lower side with the help of sellotape. After taking that leaf out, we remove that black strip and put that leaf in the water bath. We boil the leaf in water bath for 2 minutes. Then after, we put the leaf in the alcohol in a test tube in the same water bath and boil it over there for some time. After that, the removal of leaf from the beaker takes place, and what we do is, with the leaf we place it in water again. When we put in water, the leaf which has become very soft will become little um, brittle, and now it will be capable of showing the iodine test. We just put the leaf in a petri dish and add few drops of iodine solution over it, which decolorizes the leaf. After the decolorization has taken place, this uncovered part did not show positive photosynthesis, or there has been no photosynthesis. As a result, no starch is formed, and therefore it did not turn, therefore the uncovered part turned blue black in presence of iodine solution, whereas the covered portion did not perform photosynthesis, and as a result no starch was found over there, and hence the area which is covered will be showing, or which was covered by the black strip will show yellow color, resulting or um, giving the confirmation that there is no presence of starch in that area, or no photosynthesis has occurred. in that area. So light is very important for the process of photosynthesis. Entire process, just in brief, you all can just see. First of all, boiling the leaf for 2 minutes, then putting the leaf inside ethanol in the same water bath and heating it again for a minute or two. After that, the leaf is taken out, as because it is completely rid of chlorophyll, it can be just dipped in water, hot water for some time, so that the leaf becomes little brittle, and then we add the starch in order to check the sorry, we do the starch test by addition of iodine in order to check the presence of starch. The uncovered part of the leaf turns blue-black. This confirms the presence of starch, whereas the covered portion of the leaf remains yellow, showing that no starch synthesis has occurred, as because that area was covered with black strip. And henceforth, the condition or the kind of strip you will see will be like this. The area which is covered is looking yellow, whereas the area which was doing photosynthesis looks blue-black. The next experiment which we do is in order to find out that carbon dioxide is necessary for green plants to carry out photosynthesis. So materials required is almost the same, but there is one thing which is new, that is potassium hydroxide crystals. The importance of this crystal is that they are capable of absorbing carbon dioxide. So at first we begin with definitely a destarch plant. And how do we destarch a plant? By putting it in dark for 48 to 72 hours, and then after we perform the iodine test. Again, the process of iodine test is the same. We make sure that the plant which we are putting is in this particular arrangement. You can see the arrangement. This is completely a destarch plant, but we have attached a beaker. This beaker is containing potassium hydroxide and or sodium hydroxide. Both perform the same activity of absorbing water and carbon dioxide from the atmosphere or from the conical flask. And as because there is a cork which is attached along with cotton wool, so and this cotton wool is soaked in lime water to absorb any kind of carbon dioxide diffusing from the atmosphere, and it is trying to enter inside the flask, so no carbon dioxide will be allowed to enter inside the flask. So that's why we have a leaf, and that leaf is completely inside an area where there is no presence of carbon dioxide. When we perform the experiment, we will be performing the experiment in this condition. In the if after putting this particular arrangement of plant for 2 hours, and then after plucking the leaf which is inside the potassium hydroxide containing beaker, we will just plug that leaf and perform the experiment. Basically, when we try to do this experiment, we always try to keep half the portion of the leaf outside, but here we are not doing that. We have just completely put the leaf inside, so that there is no chance that there will be any influx of carbon dioxide. The various steps which are performed are still the same for the iodine test. At first we take a water bath. In that we add the leaf which is plugged from the conical flask, and then after we add that leaf in water and boil it for 2 minutes. After that we take a test tube filled with ethanol and or methylated spirit and put that leaf which has boiled for 2 hours 2 minutes inside that test tube, and after that the entire test tube is boiled for some time, a minute or two. Then after the leaf is taken out from there, and by now the chlorophyll is completely removed from the leaf, and the leaf is dipped in hot water for some time so that it becomes brittle, and from there we add the iodine to the leaf. Now after addition of methylated spirit or ethanol, after addition of iodine over the leaf, we see that there is change of blue black coloration, or there is change in color. That blue black coloration helps in the understanding that as because the leaf was within the conical flask, there is no change in blue black, or there is no blue black coloration seen in the leaf, and that's why we come to a conclusion that photosynthesis has not occurred. And carbon dioxide plays a very important role for the process of photosynthesis. So that is our conclusion. Iodine is a purple colored reagent used to test the presence or absence of starch in the leaf conducted for photosynthesis process. In the presence of starch, iodine solution will turn into blue black color, and in absence of starch it will turn brown color. So what was the result which we observed? In the experimental leaf, the part of the leaf that was kept inside the conical flask will give negative test for the starch. That is, iodine when added to this part will turn into brown color. The reason for this is that the part of experimental leaf arranged inside the flask do not get the factor carbon dioxide. So that's why, because it is absorbed by potassium hydroxide crystals kept inside the flask. So photosynthesis is not present, no starch is synthesized, and hence negative test will be there for the portion of the leaf which was inside the conical flask. The leaf which is kept out of the flask will perform photosynthesis because carbon dioxide is available to it, and hence will show positive tests for the presence of leaf iodine test. Positive iodine test will be seen by the leaf. Next experiment is for the oxygen. Why is oxygen evolved, or is oxygen necessary really evolved from the process of photosynthesis? So we need this plant. This plant is known as hydrilla. Please learn the name, and when you learn the name, it is a scientific name, so please don't forget, H will be capital and it will be underlined. So hydrilla is a seaweed, and it is found in pond. It is a pond weed, basically found in the pond. And as we are aware, most of the aquatic plants, or all the aquatic plants, they require dissolved oxygen. They are capable of taking up dissolved oxygen and carbon dioxide for the different processes they perform. For respiration, they need the presence of oxygen, whereas for photosynthesis, they require the presence of carbon dioxide. So the setup is like this. I am showing you two setup. This first setup is the actual setup which you need to know, and second one we will discuss later on. The first setup is as such that we have placed the hydrilla inside a inverted flask. Now this inverted funnel is as such, and how do we fit it? Always remember that when we are taking the hydrilla, we have to fix the hydrilla in such a way that this entire small area or the neck of the funnel is completely filled or airtight with the hydrilla stem. The size of the stem should be equivalent. Once it is done, then after we take it inverted and place it inside a filled vessel or this kind of beaker, along with a test tube which is covering the head of this funnel. When we invert the funnel, a test tube with the funnel, and we try to dip this entire setup inside the beaker containing the water, at that time we see that there is change in the entire experimental setup as soon as we put it inside water, or as soon as we put it near the sunlight. So the change which we observe is that small bubbles, they start forming in the test tube. And not only that, after some time we see that the level of water inside the test tube tends to decrease. It is because of oxygen gas taking up the place at the test tube. And this is because the plant is doing photosynthesis and it is releasing oxygen as a end product, not an end product, as a byproduct. And by the end of this experiment, we come to a conclusion that oxygen is released as an byproduct of photosynthesis.